Microbiology and the Director of Pre-Med, Pre-Health Advising at the University of Massachusetts, Amherst. Dr. Webley was born in Dry Hill District, Bellasgate in St. Catherine, Jamaica, and was the first in his family to attend high school and college. He completed a Bachelor's of Science degree in Medical Technology at the Northern Caribbean University. Dr. Webley earned the Master of Science and PhD in Microbiology at the University of Mass in Amherst, with expertise in immunology, infectious disease, and host pathogen interaction, with a concentration in vaccine development. He has received the Manning Prize for Excellence in Teaching at the university, the Black Student Union Award for Outstanding Devotion and Dedication to the Profession. Dr. Webley is a Fulbright Scholar, a recipient of the Distinguished Teaching Award in recognition for outstanding teaching accomplishments. He's also a recipient of the Commonwealth Honors College Faculty Lecture Series. He's an awardee as well as a presenter. In 2013 and 14, he served the UMass community in various capacities, including as a faculty senator, a member of the Rules Committee and the University Health Council. Dr. Webley also serves on the editorial boards of the International Journal of Vaccines and Immunization, Medical Microbiology and Diagnosis, and Microbiology and Experimentation. He's a member of the Journal of International Society of Vaccines, American Society for Microbiology, and the Chlamydia Basic Research Society. Dr. Webley's research focuses on infectious disease mechanisms and the role of specific infections in chronic diseases. Specifically, the Webley Lab is pioneering work in chlamydia vaccine development and the role of pathogenic microbes in asthma initiation and exacerbation. His laboratory was the first to culture chlamydia from bronchial velar lavage fluid, samples taken from pediatric patients with chronic severe asthma, and has since shown that early life chlamydial infection increases the risk for asthma onset and results in a unique asthma phenotype. His recent work has demonstrated the efficacy of antibiotics in treating a subset of severe asthmatics. His work has been published in reputable international journals and has made significant contributions to the field of microbiology, vaccinology, allergy, and immunology. Dr. Webley was recognized in national media coverage for organizing and moderating a panel on the 2014 Ebola outbreak that originated in Guinea, Liberia, and Sierra Leone, and spread to several other countries, including the United States. He has made multiple presentations for various groups on the coronavirus pandemic, including radio shows, television shows, YouTube and Facebook Live events. Having personally benefited from the remarkable generosity of excellent mentors, Dr. Webley is a staunch advocate for effective student mentoring, excellence in educational pursuits, and is a leader in Student Success Initiative. In his current role as Director of Pre-Med, Pre-Health Advising, he guides and he mentors students in order to enhance their competitiveness for admission to medical, dental, physician assistant programs, and other health profession programs. It is my delight and pleasure to have Dr. Webley here with us this evening, and I now turn over the time to him as he speaks to us about the COVID-19 vaccine its nuances, its efficacy, and its safety. Dr. Webley, thank you.
So, Walid, thank you very much for having me. Thank you, Ashley, for arranging this, along with Mr. Mears, someone I have really, really deep respect for. Thank you, everybody. Good afternoon, church. Thank you for having me. It is my hope that I'll be able to answer some of your questions this evening, to be able to show you a few things that hopefully will clear up maybe some misunderstandings that you might have about this current coronavirus outbreak. And maybe we can have discussions and things that I can learn some from as we go through this process. I'm going to get this panel out of your way. And you should be seeing my screen now full. Ashley, give me a thumbs up. Good. There you go. All right. So it is good to be with you. I have some slides, and depending on how I'm feeling as I'm going through it, I'll do more or less as I go through. So hopefully we can benefit maximally from our time together. So you would literally have to be under a rock not to know that we're living in a time of a great pandemic. But the truth is that pandemics are nothing new. From as far back as we have recordings of anything, we see that there have been pandemics killing some proportion of the population. And the earliest recorded pandemic, right around 165 thereabout, was the Antonine plague. Some believe that it could have been smallpox or some other type of disease. And as you come down, you see everything from syphilis to smallpox to the plague, bubonic plague, pneumonic plagues coming down. And then you get to right around 1918. And in 1918, there was a flu pandemic that was widely known as the Spanish flu. And that killed anywhere between 40 to 50 million people. This coincided with the World War. And because people were going off to war, and most countries that were involved in the war did not want their population to become disheartened by just the sheer number of people who were dying, they underreported it. Spain, however, who wasn't a part of the war, reported their full numbers, so people believed that the pandemic was worse in Spain, so it became known as the Spanish flu. And then, of course, you have HIV, which is also a pandemic. We kind of forget about it because, you know, we don't see people just dropping every day dying from it. And of course, that has led us up to this point in time where we are with the current pandemic. This is a 2018 outlook from the WHO, the World Health Organization, and it lists about 10 diseases that we see as emerging or re-emerging in nature, besides the usual suspects like Ebola and Zika. And if you notice, SARS is there, but they had what they call disease X. In other words, something that we haven't seen yet that has the potential to cause significant death and morbidity in the population. And this disease X, some people now feel that is what COVID-19 is. I don't know that I believe that this is disease X, because in my opinion, there are worse viruses out there that if they have a chance to get in the population will wreak even worse havoc than what we're currently dealing with right now. So I think somebody has the ability to write on the screen. People are trying to maybe zoom in. So there is some writing on the screen, so you have to be thoughtful there. Let's see if we can erase this. Just give me a chance here. All right, we took care of that. I know that some of the lettering is a little bit harder to see, so don't worry about it. You can ask me about it, and I will definitely go back and tell you about it. So the World Health Organization has said that the two public health interventions that have had the greatest impact on the world's health are clean water and vaccines. Hygiene, the clean waters has prevented disease after disease after disease. All of our intestinal-borne type pathogens, from Salmonella to Shigella, E. coli, so many different viruses have been prevented because of clean water, running water to wash your hands. So that's a major, major intervention overall that we have certainly benefited from. And so don't try to see these writings here. I just want to show you something. The larger circles that you're seeing there are signifying that the outer rim is 700,000 cases of these different diseases. And this one in the middle here is measles. You know, you have chicken pox, you have mumps. And essentially what you see are these large amounts of cases. And then you see this orange circle. That's when a vaccine was made and distributed. And as you can see, for every single case, from polio to diphtheria to measles to hepatitis to pertussis, all of them, the cases went down significantly the moment a vaccine was made against these diseases. So what it's really showing is that vaccines are really, really important. And so essentially what you have then, here is a great case of how vaccines work. These two boys, same ages, they were exposed to the smallpox virus on the same day, at the same time, from the same person. And the only difference is that the one on your right was vaccinated. The one on the left was not. Vaccines work, I guess, is my point. And it has been proven over and over and over again to do that and to do that effectively. So let's get back now to the current situation that we have. Coronaviruses are not new to the human population. We've known about coronaviruses for a long time. However, the vast majority of coronaviruses up until 2002, 2003 caused the common cold. You would sniffle a little bit, a little runny nose, maybe a little fever every now and then, but nothing that you would think about, nothing that you would pay a second attention to. And so if you notice, they had no fancy names, 229E, OC43, HKU1, NL63. These were the names that we gave to these coronaviruses that were discovered mostly in Asia. University of Chicago researchers discovered this family of viruses in 1965. And it has been ongoing. But then in 2002, a virus popped up that caused what we call at the time severe acute respiratory syndrome, or SARS. And the virus that was responsible for it, they called it the coronavirus 1. And it infected about 8,000 people worldwide and killed about 700, just over 700, nearly 800 people. So that's about a 10% mortality rate. And at the time, everybody was scared. Initially, nobody knew it was transmitted, and it was very problematic. And at that time, and this is really important, at that time, they actually started projects to make a vaccine against this virus. However, the pandemic ended really abruptly, and as a result of that, they couldn't complete a vaccine because you need to have an ongoing pandemic in order to make a vaccine. And people kind of forget SARS. It was in the textbooks. We talked about how it just popped up and then went away. And then in 2012, another coronavirus family member, MERS, Middle Eastern Respiratory Syndrome virus outbreak, happened. And this one was more deadly. Its mortality rate was 37%. The good part about this, if you can find some silver lining, is that it has been very sporadic, and it only exists right now in any great numbers in the Arabian Peninsula because it's transmitted directly from camels to humans. And we haven't had any real human-to-human transmission at this time. What is common among this family of viruses is that they come from some reservoir that's rodent-based. They're coming mostly from bats and rats or some other type of rodent. Most of the times they have an intermediate host. In other words, they get transmitted from the bat to some other animal, whether it's a camel or a pig, or for the SARS-CoV-1, it was a civet cat, you have from cows, you name it. And then somehow those that actually infect humans get over into the human population. And so it is believed that the current pandemic virus, SARS-CoV-2, originated from bats. That's what the genetic sequencing tells us. The bats are the reservoirs. In other words, the bats don't get sick, and that's the thing with these viruses. Bats don't get sick. Some of the worst viruses you can think about, including Ebola, actually come from bats. They are the reservoirs. The problem with bats is that they're like us. They like to be communal. They live close to each other, which means if one has something, they easily transmit it to the others. The second thing with bats is that they can fly. So they can cover great distances in a short time, and as a result of that, we actually get exposed. The intermediate host is not really known. However, what the genetic sequence shows us is that this animal called a pangolin, that is very common in China. They use the scales to make ritual potions, and so it's very expensive. People catch it, get the scales, and then they sell the meat. It is believed that this is the intermediate host. They're still investigating this, but the current coronavirus has a 97.4% sequence homology, in other words, similar genetic sequence to the virus that's commonly found in the pangolins. And somehow it could have been some other intermediate host as well, but it somehow made it into the human population and has been found to be very easily, readily transmitted. So by now you've all seen this cartoon figure from the CDC of the coronavirus. Okay, so my screen sharing just stopped. I'll pick that back up. Okay. It's telling me that my screen sharing has stopped, Ashley. Let me try one more thing. Let's go back. I think we'll get it back. Screen sharing up. How's that? Good. All right. So you see this cartoon of the virus, but what I'm showing you over on your right is actually a real electron micrograph. That's how we see it on the microscope. And you can see why it's called coronavirus, because you can see these spikes around it. And this is these proteins that you're seeing there, these spike proteins. And that's why it has the name corona, because corona means crown. So it's like it has a crown around it. That's what you're seeing there. And so this coronavirus, the disease is called COVID-19, which is coronavirus disease 2019. And then the virus itself is severe acute respiratory syndrome virus 2. That's the SARS-CoV-2 that causes this. And they named these viruses from an international society. So it's believed that this virus originated in Wuhan, China, a big bustling city. That's an international hub. People flying from all over the world, flying into China, typically flying to Wuhan. And so this is Wuhan. You can see it's a massive city. And what is very common in Wuhan are these wet markets. And they're called wet markets because they sell just about everything. So in the early 1970s, China had a really big hunger problem. They couldn't feed their people. People were dying from hunger. And as a result of that, they actually went ahead and they allowed people to start getting food by catching their own food, their own wildlife, and eating it. And then people started making an enterprise out of it, where they started selling their leftovers to people or trading it for something else. And this ballooned into what we now know as these massive wet markets where you think about it, it is sold there, from your fruits and your vegetables to meats of all types. And that is the type of situation that you find yourself. And just to give you an idea, there's no regulation there. So here you have a live trussed chicken sitting on top of a vegetable basket. Just below it, you have these live frogs for sale. Just above that, you have the cutting board where these frogs and chickens are slaughtered, and if the customer so desires. Then over here, you have some fish and frog remains. This is where he's cleaning that up. You can see the frog being slaughtered here. And essentially, all of it is done in the same area. He has a pair of gloves on, but of course, the gloves are just to protect his hand. It's not to prevent anything. Here is the unwashed buckets that they use. Here's a scale up here that they use to weigh what they're selling. So in other words, I hope I'm giving you the idea that this is a mess, right? And it's very easy to sell stuff there, and it's very easy to catch anything there. Among the animals and meats that they sell bats. People eat bats as a delicacy there. They make it in a soup, they dunk it and eat it. And so in December, and about December 12th of last year, they started noticing some cases. And it was 1 year ago today that the first report of pneumonia from this new disease came about. And what you see here is that from that time until now, we have over 80 million cases, 1.7 million deaths worldwide, with just over 330,000 of those in the United States. The United States leads all countries in terms of total number of cases of this coronavirus. Now, a lot of people talk about viruses, and it's kind of hard sometimes to conceptualize these microbes. Here you actually have a cell. The authors didn't say what type of cell it is. It looks more macrophage-like, but it could be any cell. Because it's dying, it has these blebs, these large round blebs. But all of these tiny particles that you see attached to it, these are the viruses. This is the coronavirus. This is your SARS-CoV-2. And as you can see, these viruses, multiple of them, hundreds of them, thousands of them, could get into one cell and destroy that cell. And over and over and over, they do that. And as they do that, the immune system kicks in, you get the fever, you get the other type of respiratory illnesses that people experience with this. So when you think about a virus, you can't think about it as being static, like the cartoon that I showed you. These viruses have membranes, and the membranes are very fluid, like this, so they're moving around. Those spike proteins that you see, they're also not static. It's a trimer. It's actually 3 proteins that make what is the key that unlocks our cells to get in. This is a really important concept. This virus has this protein on its surface. Its surface is littered with them, as you can see here. And that is what it used to bind to our cells. Our cells have this ACE2 receptor that it binds to and enters the cell. So here you see a cartoon showing you the spike proteins here. And here's somebody probably coughed, release these viruses. They're inhaled by somebody who's not wearing a mask. They go down into the respiratory tract, down into the lungs, into the alveolar cells that have copious amounts of these ACE2 receptors in blue here. The spike protein on the surface of the virus, those red proteins, now attached to this, bind and pull the virus close, and the virus releases this messenger, its RNA. This is its nucleic acid. This is what has all of the information to make new viruses into the cell, because it's an RNA virus. And inside of the cell, this RNA replicates. In other words, it copies itself and makes more RNA. But it also takes the RNA and make the proteins that it codes for. And all of these proteins that you see are being made by this factory inside of the cell cytoplasm called a ribosome, now results in the packaging of new viral particles that are able to exit the cell, go now back into the airway when you breathe, speak, cough, sneeze, it releases these viral particles out into the airway where new people can now become infected, right? So it's a regular process that you have happening there. And so here again is a computer animation of these spike proteins. And again, what I want to nail in your head over and over again, because I'm one of those teachers who believe that anybody can learn biology, right? What I want to get into your head over and over again is the fact that these spike proteins are the keys that the virus uses to unlock our cells, because its goal is not to kill anybody. Its goal, like all other infectious microbes, is to get into our cells, into an environment where it has food and it has the right temperature to make more of itself. It's interested in progeny, right? So you might have heard that the virus has been mutating. And you know, the several countries have blocked travel from England, from the UK, because of these mutations that the virus is having. Remember I told you that the spike protein, so this is a spike protein, it's a trimer. In other words, there's 3 different proteins come together to make this key. And you're seeing the different colors here representing the trimer. This is what it uses to get into the cell. Where you see those black dots, that's where these mutations have developed. And so this new lineage, this new variant of the virus is this B.1.1.7, not a very catchy name, but essentially what it is that the virus has somehow developed these mutations, and especially these that you're seeing right here, they are able to help the virus to actually bind more tightly to our receptors. And that's why it's more readily transmitted from one person to the next. And so with this going on, with all of what I just told you, the transmission of the virus, the fact that so many people have died from it, scientists set off on the largest vaccine project that was ever conceived, that the White House called Operation Warp Speed. And to date, we have 2 vaccines that have been released under emergency use authorization in the United States. One made by Pfizer-BioNTech. BioNTech was a company that was looking at making vaccines through the mRNA platform, same as Moderna. And so Pfizer is a big drug company. We use drugs from them all the time. And they partnered with BioNTech, this German company, to make vaccines. Moderna had already had a collaboration with the National Institutes of Health, and they partnered with the NIH to make a similar vaccine of the similar platform. And so there are over 200 vaccines right now that are being developed all over the world by various companies, and they're using different techniques. They're using different what we call platforms to develop these vaccines. So for example, the polio vaccine is what we call a whole inactivated vaccine. In other words, they take the polio virus, they make it so that it doesn't cause disease, but it's still alive, it can replicate, and they inject it in you, right? And that is how your body now responds to it and gives you immunity. So that's one type of platform. You also have platforms where the microbe is killed, but it's a whole virus. And that's how the MMR vaccine, your mumps, measles, and rubella, that's how those vaccines are made. So these are what we call platforms, right? Your human papilloma virus vaccine is a virus-like particle that they've encapsulated with it. Your flu virus is what we call a subunit vaccine. In other words, they're using pieces of protein that they're injecting into your arm. Your body sees it, realizes that it is foreign, and makes a response, an immune response against it. Because the idea of a vaccine is to prime us so that we're aware that this virus exists and make up a defense so that when we actually see it, we can respond right away. Because the immune system, the adaptive part of the immune system, that is what we need to protect us from viruses like these, takes about 2 weeks after initial infection to get up to a level where it is actually very effective. And that's important. It takes about 2 weeks, 14 days or thereabouts, before the average person makes a strong response. And so what you want is that you don't want to have a virus like this that is so contagious sitting around waiting for 2 weeks to happen. By that time, you could be dead, right? What you want is for the body to be able to respond to it right away, and that means vaccination. So at this point in time, these are the vaccines, numbers of vaccines that are in the pipeline. There are 44 in phase 1, 21 in phase 2, 18 in phase 3, 5 with limited use in different countries, and these typically include China and Russia. There are 3 vaccines approved, even though there are only 2 in the United States. There's 1 vaccine in addition to the Mrna and Pfizer vaccine that have been approved. Russia actually has this vaccine that they call the Sputnik 5 that has been approved in Russia, and they're actually using it in other countries like Argentina and other countries have latched on to this. The Russia has said that the efficacy rate is about 91.4%, which rivals the Pfizer and Mrna vaccines. 1 vaccine was pulled. This was an Australian vaccine. And this 1 pulled vaccine, in other words, they're not going to make this vaccine anymore. And it wasn't because of what happened to people as they gave it. It was because in an attempt to stabilize the spike protein that I showed you, they're trying to make it and stabilize it. They actually made a protein attachment to it that makes it look like people who get it are actually HIV positive. In other words, they test HIV positive. They don't have HIV, but the antibodies that are made are so closely related to HIV proteins that they test HIV positive. Of course, you don't want that. And so it was abandoned. So over the past week, what has happened is that Iran has another vaccine that has entered phase 1. Canada approved Mrna. Altimmune entered phase 1. So I'm just trying to show you that while we have 2 vaccines right now that are in operation, in a very short time, we could have 5 or so more vaccines around the world that are being developed and get approval. And so at the very top, you have these 3 vaccines right now, but CanSino has a vaccine that they're using this viral vector platform. Johnson & Johnson has a vaccine as well that's in phase 3. So all of these vaccines that you see on this page are in phase 3 clinical trial. This is the final clinical trial phase. So there are lots of vaccines out there. So these different vaccines are on different platforms. However, up until this point, we'd never had a vaccine developed in a year. It's unprecedented. In fact, before this year, if you told me that you could develop a vaccine in a year, I would say, come on, that's never going to happen. As you can see, these are different vaccines. Here's typhoid fever. You can see when the bug was discovered, and it took well over 100 years before a vaccine was developed. For others, 50 years, 25 years, 30 years. You can see that it takes a very long time to develop vaccines. But here's a timeline for your Pfizer vaccine, that the sequence of the virus was released in January, and within 45 days, they had already put a vaccine in somebody's arm for phase 1 trial. And we're ready to start testing this. And there's a reason for that. There's a reason for that. And however, because of how quickly the vaccines have been developed, and because of the politicization of both the pandemic and the vaccine, there are a lot of people who are hesitant to take this vaccine, which is a problem. No matter how good the vaccine is, vaccines don't work until they're put in arms. Until you're injected with it, it won't work. And what we need, especially for this pandemic, is that we need about 60, probably more than that, about 75 to 85% of the population to be vaccinated before you have what is called herd immunity, before you actually have this overall protection, where the vulnerable in the population who can't get vaccinated for one reason or another can actually be safe because they have so much of the population that's already been vaccinated. Now, the 2 vaccines that have been approved are what are called mRNA vaccines. And up until this point, they had never been used in a vaccine before. And that's an important little caveat here. And it's an important thing to think through as we're considering these vaccines. So I must step back and teach you a little bit of biology for those who don't remember this from school. You have DNA inside of your cells. And DNA lives inside of the nucleus of your cells. Your cell has two compartments, a cytoplasm, kind of the fluidy part, and then you have the nucleus. The DNA is so important to the cell that the cell will not allow the DNA to leave the nucleus, because if you alter the DNA, then you alter basically everything that the cell is making. So the DNA stays in the nucleus, and what the cell does, when a protein needs to be made, because what you're really trying to get to is proteins. Proteins are the workhorses of the body. Pretty much everything that does anything important in the body is a protein. So for example, digesting your food, that's a protein. Carrying oxygen, your hemoglobin, that's a protein, right? All of these are proteins. Your clotting factors, proteins. The molecules in your body that carries your nutrients around, like albumin, these are proteins produced by the liver and other parts of the body. So proteins are what make up the structure of your cells. These are proteins. So what you're trying to get to are proteins, but these proteins are coded for on your DNA. And so when the cell needs to make a particular protein, it copies it, that section of the DNA, because different sections of the DNA are what we call genes. So let's say, for example, that for New Year's, I was born, my birthday is on New Year's Day. And let's say that you wanted to make me a very special pie for my birthday, because you saw me and you liked my sweater that I'm wearing, and you said, let me make Wilmore a nice pie or for his birthday, and I'm going to ship it to Massachusetts to give him so he can be happy. However, the special pie that you want to make me, it's in a recipe book in your local library. However, it's not in general circulation because it's a rare recipe book that was written in the 1800s by former slaves who had come up with this fantastic recipe. So it's in this book, but it's on reserve in the library. So you go to the library, but you can't walk out with the book because the library only has one copy of it, and it is so rare that the library will not allow it to leave the library. Anybody ever taken anything off reserve? You have 2 hours in the library to go through it, and then you have to take it back to the reserve desk. So what you do then is that you don't want to sit around in the library trying to memorize the recipe, so you make a photocopy of it. Not of the entire book, because you only need one recipe. You're making me one special pie. So you make it of the pages. Maybe it's 2 pages, maybe it's a page that is on, and you make that copy. That copy is your RNA, your messenger RNA. And that's what you walk out of the library with. And you now go to your kitchen, which in terms of what we're talking about is in the cytoplasm of the cell now. And your kitchen is that protein making machinery called a ribosome. And in there is where you read the recipe line by line, item by item, and you put together and you make this cake. So this is the process that biologists call transcription translation. The transcript is a copy. Translation means that you're reading off the instructions on there, and that's what makes a protein. So let me just show you how this happens in a really highly stylized animation done by Cold Spring Harbor that I really like. And there are going to be terms in there that you don't understand. Don't worry about that. Just remember what I told you and find it in there. All right, here we go.
The DNA double helix contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, so that was a little bit of biology, but it's cool biology. So essentially what it is showing you is how you come from DNA that stays in the nucleus, never leaves the nucleus, to where you get protein that is being made, that the DNA codes for, and that is in the cytoplasm. It takes it to that big protein factory called the ribosome, where it reads, and it said it there, I'm not sure if you got it, but it reads this genetic code in what is called codons, 3 letters at a time. Because you see, the DNA is really a code. It has an alphabet. The DNA is an alphabetic code, A, T, G, C. These are called nucleotides. 4 letters is the entirety of the DNA genetic code. And the combinations of them, how they're coded, determines which amino acid gets added to the protein. Because proteins are made of amino acids, right? And these amino acids, there are 20 amino acids in nature, and those 20 amino acids are different codes from the codon, from the gene, so that they get translated in. Okay, why am I telling you all of this? Because the 2 vaccines, the Mrna and the Pfizer vaccines, are mRNA vaccines. And so these had never been tried before. So remember I told you about the spike proteins? What scientists did the moment that the gene was sequenced, the genome was sequenced for the coronavirus, SARS-CoV-2, is that they looked and found the gene that codes for the spike proteins. And once they got that, they synthesized the messenger RNA for that. In other words, the instructions of how to make the spike protein. And that's what's in the vaccine. The vaccine contains the messenger RNA, the message that will make the spike protein, not the whole virus, just the spike protein. And it's enveloped in lipids, in fat. So they just put it in a fat molecule, surrounded by fat. And so you can imagine that there's several copies. So instead of just having 1 photocopy, you probably make 100. And that's what's in this lipid here. That gets injected into our arm, into muscle cells. Muscle cells are known to readily pick up genetic material. And so inside of the muscle cells, these get picked up, and that ribosome, that big protein, blue protein blob that you saw, that is in every cell, that reads the instruction on this synthetic piece of messenger RNA that got injected into us, and it makes the spike proteins. The spike proteins now get released from the cells that are making them. They also get picked up by immune cells, which now break them down and present them to other immune cells, and you now make this Y-shaped protein called an antibody. And these antibodies are your savior. This is why vaccines work. Because these Y-shaped molecules, they now know this key, which is what the coronavirus uses to get in. And what they do is that they actively bind to that key and block it. It can't fit the lock anymore. It can't bind to that ACE2 receptor on our cells that it used to get. And that is why we're protected from infection with this virus. So when the virus comes in now, here's a virus. It has all of these Y-shaped proteins binding to all of these spike proteins and blocking them, preventing them from getting it. It's like taking somebody's key and wrapping duct tape all around it. And when they go to the door to put the key in the door, it just doesn't fit anymore. Right? And so that is essentially what is happening here. So this is how the messenger RNA works. So people will tell you, oh, you know, this messenger RNA that they're giving in the vaccine is going to alter your DNA. Can't be true, right? Because DNA and RNA are two different things. The DNA is in the nucleus, the RNA is in the cytoplasm, and the RNA can't make it into the nucleus. You don't have any reverse. It goes in one direction. All right, so let me just play another animation for you.
mRNA technology has never been used before in an approved vaccine, but scientists are using it now to combat COVID-19. mRNA, or messenger ribonucleic acid, are snippets of genetic code that tell cells to build proteins. mRNA vaccines take advantage of that protein-building process to trigger an immune response and build immunity to SARS-CoV-2, the virus that causes COVID-19. Instead of using a small dose of live or dead virus like some other vaccines, mRNA vaccines use a tiny piece of the virus's own genetic material. Here's how it works. This is the virus. These are called spike proteins. Scientists have isolated the genetic code for those proteins, a set of instructions that are put into the vaccine so your body knows how to make them. Your immune system won't make an actual virus particle, but it will make copies of the angry red spike protein. In other words, no gray ball, but an angry red spike. Once vaccinated, your body starts making those spike proteins so your immune system will develop antibodies to fight the real thing, SARS-CoV-2, the virus that causes COVID-19. The Pfizer and Mrna vaccines that are created for COVID-19 will be the first licensed mRNA vaccines ever.
Okay. So that is what we have for a vaccine. I'm going to skip a few things here and just probably address a few of the myths about the current vaccine and coronavirus. So the first myth is that the new coronavirus was deliberately created or released by people, government labs. Simply not true. The genetic sequence shows that these are very closely related to what you have in the bats in the wild and the pangolins. So we know that when people create genetic things, it leaves a genetic signature. We only have so much repertoire. It's very easy to find that. The other myth is that the vaccine was rushed. Now, I told you that this vaccine was developed in less than a year. However, it wasn't rushed, and here's why. Because of the new platform, the messenger RNA platform, never used before, but now is being used. If we were developing a vaccine by the natural, normal way that all the rest of vaccines that we have are developed, you would have to make that protein, you would have to purify that protein by growing up tons and tons of the virus, very dangerous, in level 3 facilities. And you would now have to get this purified protein, get all of the purification chemicals out of it, and then you would formulate it and inject it into somebody with what is called an adjuvant to enhance the immune response. That takes a long time. Number two, because we're in the middle of a pandemic and the government has thrown so much money at this, they overlap different phases of clinical trials. That has never been done before because no single company has that type of money, right? Only federal money could give you that leverage. So essentially, a phase 1 clinical trial is where you have less than 100 people that you injected in at different doses. You see if it's safe. Phase 2 clinical trial is where you look for what we call efficacy. Does it actually work? Does it induce antibodies, and is it still safe, right? What the government did is that it gave these companies enough money so that they could run a phase 1 and a phase 2 trial at the same time. And then once they got phase 2 data, they could run a phase 3 trial at the same time that they're finishing up the phase 2. but also start manufacturing the vaccine while they're in phase 3. Now, you might ask, what if the phase 3 didn't work? Well, they'll just throw it away and do something else. Why? Because you have this limitless amount of money. You couldn't do that if you were a Moderna or a Pfizer or a BioNTech in a regular time. It just would not happen. Number 3, the reason why the vaccine was able to happen so quickly is because typically you don't have so many people getting infected in any pandemic that we've ever had. Because you need people who are getting infected to try the vaccine, because what you're doing is that you're taking people, you're giving half of them a placebo, or you're giving some of them a placebo, and the rest of them you're giving the real thing, the vaccine. And so essentially you're doing this, right? You're giving some people the vaccine, some people the placebo, and you're waiting up to see who gets infected. So they're randomly distributed. Neither the person giving the vaccine nor the person getting it know if they're getting the placebo or they're getting the real deal. And they wait up. You have some monitoring group that waits up and they check these people on a regular basis and see when they get infected. So when they get infected, and after you have had a certain number of people infected, you're hoping that they get infected, because if they don't, you won't know if the vaccine works. And then you go and you check now and see who was getting infected. Was it more of the people who got the placebo or those who got the vaccine? And if it's those that got the placebo, how much more? And that's the statistics that we get. That's what the FDA uses to give us authorization for the vaccine. So without a raging pandemic like the one we currently have, you would never be able to do this type of testing because you would just not have enough people. The other myth is that the vaccine will edit your DNA. I just showed you that that just doesn't happen, right? Then the vaccine causes infertility in women and sterility in men. Just not true. None of the data has shown this. In fact, while they did not deliberately give the vaccine to pregnant women, there were several women who got pregnant after the first and after the second dose of the vaccine. So we know that people are not losing their fertility. That the vaccine can cause COVID-19 or reversible adverse events. Just not happening. The adverse events that we're seeing, even those that are anaphylaxis, they're reversible. People get allergies all the time. That you can use vitamin D and other supplements and that will protect you from getting the virus or treating it. Not true. While it is true that you need to eat right and we need to exercise and be at our very best and be as healthy as we can, that's how you know you have the best immune system. The truth is that we don't see anything out there today that will prevent you from getting infected. And it doesn't matter how much Cersei you're gonna boil, my Jamaican friends, and how much Search Me Hot, and all of those things, it does not prevent the virus from infecting you. That the pandemic was invented to cover up the effects of 5G radiation. Radiation is an electromagnetic wave. It is not a biological entity. This virus is a biological entity that is causing the disease. 5G does not penetrate, doesn't do anything to us in terms of our cells. And then finally, that wearing masks increases the amount of carbon dioxide you breathe in, and it will make you sick. And as a result of that, you shouldn't be wearing a mask. Simply not true. Surgeons have been wearing masks forever. They wear it throughout, you have surgeons who are in surgery for 18, 20 hours wearing a mask. You see, carbon dioxide and oxygen, they're gases. They readily diffuse through the cloth mask or the surgical mask, even the N95 mask that people wear. You're not gonna pass out because you're wearing that. And I'll stop there and we can have some discussions now.
Wonderful, thank you, Dr. Webley. We have a few questions. The first one says, what proof do we have that these are truly myths, considering we don't have that longitudinal study?
Because there's nothing here that I'm saying that requires us to have longitudinal study to show. We need longitudinal studies to show us how long the vaccine protects for. That we don't know yet. We don't know if this vaccine will protect you for 6 months, for 8 months, for a year. We have no idea. However, If you go back to the FDA database and you look at all vaccines that have ever been made and issued that we take, the most significant side effects or adverse events take place within 2 months of the second dose of the vaccine. Remember that there are people who have been getting these vaccines, both Moderna and the Pfizer vaccine. The people who were in phase 1 were getting this since March, right? We like to think that this vaccine is something that just popped up. They're individuals, lots of them around, thousands of them, who have been getting these vaccines way more than 6 months, right? So we know that should not be a factor. For the people who have adverse events from this vaccine, having a sore arm, having a fever, even for those who have anaphylaxis, these are short-term effects that go away after a relatively short period of time. Although these are new vaccine platforms, you should remember, or you probably should know, that the technology is not new. I told you that they had been thinking about making a coronavirus vaccine since the first SARS in 2002, 2003. Since then, they've been trying to develop this. But also the mRNA platform, Moderna is a 10-year-old company. And that is all that Moderna has ever done. Every medicine, everything that they've tried to develop has been messenger RNA. BioNTech have been developing an mRNA platform as well. So the technology for this is literally like a plug and play that has been in the process for a long time.
Okay, thank you. Our next question says, has anyone died from any of these vaccines, even the ones that were abandoned in Australia?
No.
Okay. Our next question says, what type of vaccine did President Trump get?
President Trump hasn't gotten a vaccine. He hasn't taken one. But I think Mike Pence, some of the others got the Mrna vaccine, Fauci got the Mrna vaccine. President Trump got infected with COVID and was treated with a cocktail of medications. Many of them were in phase 1 trials. So far, at least up until last night, as far as has been reported, he hasn't yet gotten the vaccine.
We have a question that says, as far as we're concerned, the virus is still not completely understood. If the vaccine is to protect us, why do we still need to wear masks and social distance?
Very good question. Excellent question. And it's for that very reason. So number one, we know that the vaccine significantly reduces the ability for the virus to cause disease. And that's what vaccines do. You getting a vaccine doesn't prevent you from getting infected. It prevents disease, right? In other words, you do still get infected, but exactly what I show you happen, that the viruses get neutralized, the vast majority of them, and they get taken out. And so you don't expect people to have to be hospitalized for this. However, 95% of people are protected in that way. You still have 5% of people who get vaccinated and still have adverse events, right? Number two, we don't yet have herd immunity. In other words, there's still lots and lots and lots of people in the population who are susceptible to the virus and who can get infected. And so the virus, while the virus can give you this 95% protection, wearing a mask and social distancing and physical distancing and washing your hands, that's going to give you 100%, right? So you still have to wear it. Number three, we do not know if somebody who has gotten the vaccine can actually transmit the virus to somebody else. That is still in trial. We don't know that. That's an important part of what is going on now, of what they're testing. Because you could get the vaccine and it significantly reduced the number of viruses to the extent where you're showing no symptoms. It does not mean that you don't have viruses in your nostril. It does not mean that you're not coughing and sneezing, and when you're talking, they're there. And if that is the case, if there's enough, you could actually transmit it to somebody else. The fourth thing is that there are demographics of the population who cannot be vaccinated because the vaccine wasn't tested on them. For the Mrna vaccine, anyone below 18 years old. for the Pfizer vaccine, anyone below 16 years old. They're now in clinical trials testing ages 12 to 16, but we don't know yet what that dose needs to be and how it needs to in other words, there will still be, even after everybody who's currently eligible for the current vaccine has been vaccinated, there'll still be a ton of people who are susceptible, who cannot be vaccinated under the current emergency use authorization.
Our next question says, you mentioned that the drug companies did not have unlimited cash for development of the vaccine. However, Pfizer did not take government money. Can you explain the difference?
Yeah, so Pfizer didn't get the type of money that Moderna got, for example. But Pfizer got upfront money from the federal government that paid for specific numbers of doses of the vaccine, right? They use that money now to go into research and development, right? So it's kind of a nuanced explanation of that. Now, Pfizer is a large drug company. They have a lot of money in R&D. Moderna is a very new company, a tiny little company down in Cambridge here in Massachusetts that started in 2012. So they definitely needed that money. Several other companies have also gotten money from the federal government to make vaccines, and they're in the process of doing that right now.
Our next question asks, was the infection underrecorded in China?
It is very likely. China is known for doing this, especially earlier on in the pandemic. However, because they did the same thing in the outbreak of SARS in 2002, they actually reported virus in December 28th of last year to the local CDC. At the time, many people believed that they weren't talking about just how many. And the way we know that is that a few days later, well, almost a month later, they locked down the whole city of Wuhan. You wouldn't have locked down the whole city of Wuhan for the numbers that they were reporting at the time. It could have been that they just didn't know the full numbers, or they were being instructed by what is, by all means, a communist government to tell them not to report all the cases that they were seeing. It could also be that they were conflating cases of the coronavirus with other respiratory diseases that people were having, and that happened to some extent. But I fully believe that China didn't report, whether deliberately or not, that they didn't fully report the total number of cases they had in December, January.
I think it's Sister Jones has her hand up. I don't know if Sister Jones had a question. Okay. Next, we have a question that asks about stem cells and their use of the vaccine, if you have any thoughts on that.
Stem cells, so these vaccines, as I said, they're synthetic, right? These mRNA vaccines, the messenger RNA was synthesized through a computer program in a lab, right? They weren't grown in anything. So no stem cells are being used for vaccine. I know that people had concerns about the, I think it might have been the British vaccine, the name of it is slipping my mind right now, where the virus, the adenovirus, the host virus that they're using. So you're literally using a non-disease causing virus as a vessel to carry in the spike protein gene. And those are grown up in cells, fetal cells that were taken from an aborted fetus maybe in the 1960s, right? And that's all, you have to grow them in cells. So in my research lab, I have cells from animals, cells from humans. Typically, we use cancer cells, but there's certain studies that we do that you don't want to use cancer cells, because cancer cells by nature are mutated, right? It is mutation that led to the cancer in the first place. And there are certain tests that we do that you don't want to use a cell that has mutations. And so you want to use cells that are replicating, right? Not dying on you, because regular cells that you take out of the body die within a few replications. However, fetal cells, cells that are taken from early in a pregnancy, have much more longevity because of what we call telomeres. They have longer telomeres in their DNA. And so these were obtained legally, and they're grown up to grow the virus. But when you actually reach the stage of the vaccine, none of those cells are actually in the vaccine. You wouldn't want to do that. You wouldn't want to give a vaccine with any cells or cellular debris. And that's a part of the purification that I talk about. These are the things that take a very long time and take really sophisticated processing that actually delay some of these vaccine developments. So I know that for people who are anti-vaxxers, this is one of their common things to say, oh, you know, it has fetal cells in it, or, you know, they kill babies, they're killing babies to make these vaccines. What they don't realize is that I have a cell line. You might have heard about Henrietta Lacks. You might have read the New York Times bestseller on Henrietta Lacks, this black woman in Baltimore who died of cervical cancer in the early 1900s, and her cells were used for research without her knowledge. Well, Henrietta Lacks has been dead now for over 100 years, but I still have some of her cells in my lab that we use for research, because these cells are immortal. They keep growing and growing and growing. It's the same thing with these fetal cells. They've been grown over many generations and look nothing like what they did when they were initially taken out.
Thank you. We have another question that said, if you recently had COVID, should you wait to take the vaccine? And also, we have multiple questions asking that if you've already had the virus, do you still need the vaccine?
Yeah, so this is an interesting point. So what the CDC recommends is that if you have had COVID, that you still take the vaccine. And the reason for that is that study after study has shown that only a percentage of people who actually get infected with the virus actually have what we call neutralizing antibodies. In other words, have an immune response that will protect you from the virus. I've had students who got infected in the early part of the pandemic, in March, April, May, thereabouts. And when we tested them for the antibodies, for the immune response to the virus in December, they didn't have any antibodies. Right? That is not necessarily true for everybody, but again, you have nuanced immune responses. So the idea is with the vaccine, it's been controlled. You're upping the total number of proteins. You're showing this to the immune system. You're hyper-immunizing, giving 2 doses. So that now will generate enough, not just antibodies, but memory cells, which you need. You need cells, immune cells in your body that remembers the virus, the particles, so that when it comes, you can actually protect yourself against it. So they do advise that you get vaccinated again.
We have a question asking, which of the vaccines would you recommend, the Mrna or the Pfizer?
Yeah, and so both of them have the same efficacy. And my goal here is not to recommend to you vaccines, right? I don't want to come and tell you that you should take a vaccine or not. That is something that you need to make up your own minds on. What I want to do is to cut through all of the myths and give you the real deal, to give you the correct information, the scientific information. And hopefully that's why I painstakingly, if you notice that I was repeating several things, that's how I teach. I figure if I repeat it enough times in different ways, you will learn it, you'll understand it. And hopefully if you understand it well enough, you'll be able to make up your mind about which vaccine you're gonna take and if you're gonna take any. However, for the 2 vaccines that are currently licensed in the United States, they are the same platform. Both of them are messenger RNA vaccines. They have similar efficacy, about 95%. The only difference right now is that the Pfizer vaccine has to be kept at super cold temperatures, minus 80 degrees Celsius. And the Mrna vaccine can be kept in your regular freezer that you have at home. In other words, it can reach people further away from your developed cities and stuff where you have lots of these freezers, because these freezers, as you might imagine, are not regular freezers. They cost anywhere between $12,000 to $18,000 a piece. They take a lot to keep them super cool. You have to be very thoughtful about how many times you open them per day. So for example, one of the first batches that they sent up into a remote part of Alaska had to be thrown away because the temperature dropped in transit. And of course, they have detectors on them. They detected it and had to throw it out. You don't want that to happen, especially when you're having so few doses relatively in this early stage of getting vaccinated. And so essentially what I would say is both mRNA vaccines at this point have the similar efficacy, and I would take any of them when my turn comes. people have their preferences. My wife, for example, who's on iCharm, would rather get the Mrna vaccine. She just likes them better, maybe because they're in our state. I don't know. That would be her preference. However, I think she's going to end up getting the Pfizer vaccine on Monday. So, you just have to take what you get.
One person asking, can you explain the reason for the second dose?
Very good. So when you get the first dose of the vaccine, what they found in the clinical trials is that it only gave you about 52% protection, right? Because it primes your immune system. Your immune system sees it, but it is not strong enough. The response is not strong enough to offer you the level of protection. So this 95% protection that we're talking about, this is for people who have received both doses. And for the Pfizer vaccine, this is 3 weeks after the first dose, you get the second dose. And it is that second dose that brings you up to the 95% protection after 2 weeks. Right? So this 95% protection is 2 weeks after the second dose. For the Mrna vaccine, it is 1 month after the first dose that you take the second dose, and that's what brings you up to the 95%. And so if you don't take both doses, and I mean both doses of the same vaccine, so you don't want to have the first dose of Mrna and then you get the second dose of Pfizer. That's not the way they were created. You want to take both doses of it. That's what gives you protection. Not that you don't get protection from the first dose, it's just not enough.
I think you touched on this a little bit already, but we have someone asking, are you still contagious after taking the vaccine? And if so, for how long?
Yeah, we don't know. And that's a really important part. And that's why for people getting the vaccine, the important thing is to continue wearing your mask. It gives you some level of assurance, but you wear your mask and your social distance as much as possible so that you can protect other people who haven't taken the vaccine. So again, there are going to be people who, because of underlying health conditions, maybe some types of autoimmunity, who knows, who will not be able to take this vaccine, as they are not able to take several other vaccines that are around. And you don't want to have those individuals exposed unnecessarily.
We have one person saying, it has been reported that individuals on the FDA board were threatened to be fired if they did not approve the vaccine. What is your point of view on this?
They might have been threatened, I'm not sure by whom, but these are scientists. And scientists, true scientists, don't care about threats, right? These people have been trained to evaluate data. And what I can tell you is that the same rigor, so you would have heard, if you were watching this very closely, that they got the paperwork from the clinical trials, and they took somewhere around 10 days for every member on that vaccine board to fully evaluate it, all the data that they have, write a summary on it before they met. And they met in a public way, transparent, discussed all of the possibilities and what the data was saying, and then they vote their conscience. So I don't think that any of these two vaccines got a unanimous vote, because for one of them, the age range was an important part. Pfizer wanted their vaccine to be given to people down to 12 years old, and they said, no, we want that vaccine to be at 16. And so, these are scientists. These are people who believe very firmly in the data. Moreover, all of us are seeing the data. This data is no longer hidden. I read through the entire data set myself, right? So even after the board had met and made their recommendations, I walked myself through the entire data set. This is what we're trained to do as scientists, to read research papers, to read data. The New England Journal of Medicine peer-reviewed and published the data as well. And so we know what the data is saying, and we can evaluate it ourselves. So even if people were threatened by some threat from whoever, it would have made zero difference. These are professionals who were never going to jeopardize the safety of people for anything. They're not paid to approve a vaccine. They're paid to evaluate a vaccine.
Absolutely. We have someone asking, would the vaccine fully protect against new mutations in the virus?
Yeah, that's one of our concerns, right? But there are two things there. From what we've seen so far, there's nothing that would make us believe that even the new mutation they're having in the UK would not respond to the vaccine, because the antibodies that are made, it's not a single clone of them that are being made against just one little point on the protein. This protein is a big protein, so you're having several antibodies being made. In other words, it's being blocked at several sites on the protein. And the other thing to really think about is that this virus, unlike something like an HIV or an influenza, has what we call a proofreading system. In other words, this virus doesn't make a lot of mistakes. It corrects its mistakes before it makes its final transcript. And this is unusual, but this is a large virus. And so these larger viruses can have proteins. So it's like writing in a Word document, but you have spell check on, right? This virus has a spell check turned on. And so lucky for us, because it is such an infectious microbe, that if it was making mistakes that would lead to it being more productive or having more severe disease, then we'd be in trouble. However, it is clear that the mutations that we've seen in this virus now, 6 or 7 mutations so far that seem significant, that they've made the virus be more readily transmitted. So if you have one virus and let's say its transmissibility is at 60%, and then it makes a mutation that makes its transmissibility go up now to 75%, your 75% variant is going to take over in the population because it would be more efficient at being transmitted. And so that's what we're seeing, that in a relatively short period of time, this new variant that you're seeing in the UK is taking over because it is that much more efficient than the original one. Hopefully, we won't have a situation where it would evade the vaccine. However, that's the beauty of the mRNA platform. If that happens, all they have to do is just go back and put that variant into the computer, generate a messenger RNA from that, and throw it in the vaccine. And it would protect. It's like a plug and play for the mRNA vaccine platform.
Speaking of the novelty of the mRNA vaccine, we have someone saying a number of skeptics will not take the mRNA vaccine because of its novelty. Are any of the vaccine developers working on plasmid DNA vaccine to appease the fears of these people?
Yeah, and you know, I understand the fear, and then that's why I say I'm not here trying to tell you that you should take a vaccine or not, because these are real fears, very real fears that people have. Again, I don't have those fears because I understand the way the platform works. There's nothing in there. In other words, all you're doing is that you're taking a message, you're taking the code to make one protein, and you're utilizing the protein making machinery of your own cells to make and deliver that to the body so that you can get vaccinated against it, as opposed to making it ex vivo, making it in a lab, making the protein in a lab, and then injecting the protein into an individual. That's the only difference. We're making the protein in a lab, we're doing the same thing. So here's the thing. The same thing that this vaccine is doing is exactly what the virus does. You remember when I showed you the animation of the virus? The virus injected its RNA into the cell. And what did the cell do? The cell made all the proteins that the virus wanted made. This virus is a virus that makes 30 proteins that now gets assembled with new nucleic acid in there, with new RNA in there, and then the virus buds out. In other words, what Moderna and Pfizer have done is that they've done the exact thing that the virus does, except that they're not giving the whole genome copy. They're just giving that one gene that make that one protein that we know can be neutralizing, because the virus needs that one protein in order to attach and unlock our cells to get in. So because of that, I don't have those fears. However, the vaccine that CanSino is making, the one from Johnson & Johnson, the one from AstraZeneca, all of these are using viral vectors. It's a completely different platforms that have been used to make vaccines in the past. and lots of them down the road coming up. Novavax is making a vaccine that's protein subunit. Sinovac is making an inactivated vaccine. So there are about another 40, 50 vaccines coming up through the chain that are not messenger RNA vaccines.
Wonderful. We have a question asking, would you expect to get sick after taking the vaccine? And if so, for how long and how severe would the symptoms be?
Yeah, I would expect that if it's working, you would get sick, right? That's why you get sick when you get infected in parts. Part of your sickness, part of how you feel, that yucky feeling, is really your immune system at work. That fever, that little bit of weakness and nausea that you might feel, that is the immune system working. So I would expect that if the vaccine works, you should feel a little sick. You will feel as you have a sore arm, because when you take a needle of whatever size and you push it through muscle cells, that's going to hurt. And that's why the flu vaccine hurt you, because it's the same situation. You're putting it in muscle cells. So that you'll have. Now, there have been some side effects, some adverse events that are unexplained. So for example, you've had people who have anaphylaxis. These vaccines have no preservatives in them, the messenger RNA vaccines. Again, it's just nucleic acid and lipids, no preservatives. And so for these individuals, it's not clear to me if they're being allergic to one of these lipids, which is just fats that you're putting in. It would be unusual for them to be allergic to messenger RNA. That wouldn't make sense. But again, we just don't know what they're allergic to. There were about 3 patients in the Pfizer vaccine arm that actually had Bell's palsy. And this was temporary, you know, where they had this flaccid paralysis, maybe of the face, and then it went away. What caused that? We don't know. But there are individuals who have multiple allergies, and when they get an injection of whatever sort, they develop an allergic reaction. So for these individuals, the CDC has said that they should be vaccinated in the presence of healthcare professionals as much as possible. In other words, in a hospital or clinic setting, so that they can be monitored. So if something happens, they can be taken care of right away.
We have one question asking if you could explain the use of nanotechnology in the COVID-19 vaccine.
Yeah, and so this lipid particle that I'm talking about, that the messenger RNA is encapsulated in other words, it's like you go to the pharmacy and you pick up your pills. And I don't remember which pills come in capsules, but some of them do, right? You have that little plastic capsule that contain the powder or liquid or whatever, or the gels. You take vitamins and they're in this gel form. The vitamins is encased in this gel. It's the same thing with these little lipid capsules. The lipid capsules are small, so they're nanoparticles. So that's the nanotechnology, right? These are very small, less than 100 microns in diameter. Nanoparticles that have encapsulated this vaccine, and that's what they're injected inside of you.
We have one question asking, in your opinion, after this pandemic comes to a close, what should be the first step we as a global community take so that we are better prepared for the next pandemic? Yeah,
choose better leaders. You know, in a pandemic, leadership is really important. And when people choose leaders, you can't just be idealistic, where you choose somebody to be a president, a prime minister, or something who doesn't understand how to take care of what you have and what needs to take place in something in a pandemic like this. You need to have people believe in science, because this is scientifically based. And if you have a leader who doesn't believe the science, who won't compel people, if you must, to protect themselves, then people are going to die. The United States has the most deaths from this pandemic. There's a reason for that. There are countries that got the pandemic at the same time the United States did, South Korea, for example, and proportionally. Look at Canada. Canada has less people who have died over the entirety of the pandemic than what we're having dying every day. It is crazy. So that's the first thing. The second thing is that we need to have strategically placed experts, like what you have the CDC detectives all around the world. We had some of these in China before they were pulled out. You have them all over Africa. We also need to have virus hunters. These are epidemiologists and microbiologists and virologists who go all over the world, especially close to rainforests and natural preserves where you have bats, lots of bats, and people are encroaching. That's one of the things that has happened in China. People now live in such close proximity to bats that you could readily get something from a bat urine or droppings that could cause disease. So we have a lot of things to do. This is a global community, and you can't say you're going to do it in one place and not in the other, because you see how quickly a virus can travel via airplane from one place to the next. And when you have a virus that can infect people without them showing any signs that they're infected, you can transmit that virus to somebody else. You're going to cause some serious problems. And that has to be in place. So there's a whole host of things, nothing new necessarily, things that have literally been codified and have been in place for a long time that were just not followed during this pandemic. And this has resulted in the devastation and the cost of lives that we're seeing. The other thing is, we live in an age where people believe that the rules just don't apply. We have people who are very selfish, who refuse to give up having a party, a birthday party. Or you have people, Seventh-day Adventists, who go around telling people that it's only cowards who are not going to church, and that you should go out and have worship because God expects you to be in church. And these stupid things cost lives, right? You have now Seventh-day Adventist Facebook pages that are telling people not to take the vaccine because the church and Ellen White says you shouldn't, and telling people all sorts of things, that the vaccine is going to change you. This is coming from Seventh-day Adventist people. And they not only post it on social media, they pass it around in WhatsApp groups, they send it around to people. And when you have a message that starts out, send it to as many people as possible before the government deletes it. You know it's a lie, right? That's the first clue that is just bogus, right? And we need to stop doing this. If I get something that I know is not true, I don't send it to one more person. It stops at me. We have to do our part, because you might send this to somebody and that person takes it wholeheartedly, and it could cost that person or a relative their lives. We have become callous in this regard. So what I see going on, where people have decided, I'm going to fly, I'm going to go celebrate Christmas, and I'm going to celebrate Thanksgiving, and by the way, nobody's going to stop me from celebrating New Year's Day, and you're not going to get me to wear a mask. These things are selfishness at the highest level, where we refuse to protect, we refuse to be our brother's keeper, right? This is what it's all about. Wearing a mask, physical distancing, washing your hands is being your brother's keeper. You might get the virus and you get sick and you get better and you don't die. Somebody who has an underlying condition, diabetes, heart disease, will die. And you know what? Black men are the largest casualty of this disease that nobody talks about. African American men are dying at an unprecedented rate from this virus. If you think I'm lying, go check it out. There are news reports on this that somehow just evades us. So I think we have to be very thoughtful. And that's my own little preachy segment for this evening. I'm typically very much objective.
Amen. We have a question asking, were there any medications on the market that could have been modified or used to help prevent the virus, or at least alleviate the symptoms?
Yeah, good question. You guys are so smart. They have been looking for this ever since the pandemic came around. That's how remdesivir started to be used. They went into what is called the small drug database. They looked at all the drugs that they have now, and they take the imprint of the spike protein and how the virus actually enters the cell, the different enzymes that it has, the furin enzyme that it needs to cleave. And they have looked for drugs to be able to do this. They just have not been able to find one. that doesn't mean that they're going to stop looking. And that's what's missing. You know, we talk about a vaccine. We talk about this prevention part of it. What is missing are drugs that can effectively treat people pre-hospitalization, that will prevent them from actually having to go into the hospital. And this has been sparse. We just haven't found anything that I've seen, nothing that I've read. Somebody might have read something. I just haven't seen anything in my sphere of reading that is able to do that. And that I expect that still a lot of companies are working on. I'm sorry, but I saw Mr. Rutherford, is it Patrick? He's had his hand up. I think he probably doesn't know how to put a message in. Patrick, you have a question? Let me have it. Unmute. You see, I have a 30-inch screen so I can see.
The host doesn't let me in.
Okay. What's your question?
Okay, I'm in now. Good. You mentioned, and the data I've read bears it out, that after a while, people lose the antibodies that's created when they get infected.
Some people.
Some people. Have they done any studies about memory cells, if people get it? How long the memory cells keep that information, are able to generate more antibodies if they should get infected again?
Right. So it's interesting, Patrick. So it's not that these individuals don't make antibodies and have memory cells. However, what they have the antibodies and memory cells to are parts of the virus that are not neutralizing. It's not the spike protein part. You see, one of the things I didn't point out to you in those colorful spike protein pictures that I showed is that it's heavily glycosylated. And if you know about memory cells, you probably know about a little bit about the immune system, that glycans or sugars do not induce a very strong immune response because they don't call in your T cells. It's your T cells that result in memory cells. And so the viruses do this all the time. They strategically heavily glycosylate their protein, put sugars all over their proteins, so that those parts, the glycoprotein part, is actually what's inducing most of the antibodies. But what the business end of that protein is really where you want the antibodies to be made against, that bottom part of the spike protein that directly contacts. So when I showed you one of the animations that I had, let me pop this up and show you again. So let's see if okay, here we go. Do you see that purple part right here? Yeah. It's so this one has turned now. It's the purple part right here that actually binds to the ACE2 receptor. That's where you want antibodies to be made. The rest of this fill diagram, all over these parts, have lots of sugars on them, and those will also induce production of antibodies, but not strong enough memory cells that will live, and not antibodies that are actually going to totally prevent the virus from binding. So this is the part where those purple balls are that you want antibodies to be made against, so that when the antibodies bind, that prevents that little part that goes up and down. You'll see, you see that little part that open and close? You want it to be in the closed conformation, and that's how the vaccines are made. When the mRNA is made, it's made against in the closed conformation so that you can actually prevent that binding. And that's what we're not seeing. We're not seeing in some people that type of neutralizing antibodies. And you have other parts of the virus that antibodies might be made against, but when they look for neutralizing antibodies, you're just not seeing it long term, especially in younger people who had milder disease. Okay. Thank you for your question, Mr. Ruff.
Can I just ask one more?
The host has the control over this.
Yeah, can I ask one more question?
Okay.
Have they done any studies on people who have had the disease already, and what's the rate of reinfection? Yeah, so it's one of the interesting things. We've seen a few people who have been documented to being reinfected. There have been several anecdotal stories of people who have been reinfected. But by and large, most people do not get reinfected with this virus, at least not in the short term. But remember, again, this pandemic hasn't been around for very long. You know, I know we feel like it's been a long time, but the 1919 pandemic ran for 2 full years, right? We've just completed a year, not a year yet in the United States, but so we're very impatient. We live in the microwave age,
and instant messaging age where everything should happen right away. Pandemics don't care about that, and especially when people are not following the rules. This pandemic totally loved people who love to crowd, and so it will continue to infect. But we just don't have enough time yet to determine that. I'm sure that lots of studies come out almost every day on these things. They just don't have the bandwidth yet to get the type of data that can completely. Yeah.
We have a few more questions. One of them says, for people with autoimmune disorders or issues with their autoimmune system, is this mRNA vaccine safe?
Yeah, and so they were very purposeful in this study to include people who had autoimmune diseases. And so there's several autoimmune conditions for which this vaccine has been vaccinated. So what I'm going to do, I'm going to put in the chat right now the emergency use authorization for, so this is the summary for this one that I just put in was for Moderna. I'm going to put in the Pfizer as well. And you might not be able to read everything in there, but you can see the data tables for the different groups of people. Oh, did I just, okay, I think I DM'd. Let me throw it in for everybody. So this one is for Pfizer. Let me pull up back the Moderna. So this is the 50-something page document that has in most of these data sets. It shows you the ethnic groups, the different groups of people that have been tested. That's why you have a clinical trial. You want to see efficacy and safety in every group of individuals. So you can see that they had people with obesity and diabetes and chronic lung disease and HIV. HIV is an immunodeficiency disease. And you can see all the other diseases that they had, that people had when they got vaccinated. But they were deliberate in trying to have people who had some of these conditions in the study.
Wonderful, thank you for those resources. We have one question asking, do you think other countries will require you to prove that you are vaccinated before allowing you entry?
Yeah, lots of countries are doing that right now. So I have a set of really good friends, and they went to Aruba for the Christmas holiday. They had to get a COVID test within 72 hours of departing for Aruba. Then they had to show when they got there these test results that they're negative. And while they're there, they are required to take another test. And so, lots of countries are doing almost what you would call a COVID passport, a test passport, where you have to demonstrate that you're negative. Jamaica was doing that at the very beginning, where you would get tested at the airport, get a tracking app on so that they know where you are at all times, and you'd have to prove that you're negative before you were allowed out. It's an expensive venture, but now what they're doing is that they've levied a COVID tax on people who are traveling in, so they're able to pay for this. So countries are very creative. This country is probably one of the few that in many ports you're not required to take a COVID test before getting in.
We have one question here asking, I think you spoke on this a little already, but about natural treatments like eucalyptus oil or mint.
Yeah, I mean, if those things make you feel better when you're COVID infected, I would say go for it. However, the remedies that people talk about and that they post on WhatsApp and YouTube and all these things, they're misleading. Because the fact that a friend of your great aunt had a friend who got COVID and they used the eucalyptus oil and took their vitamin D and they got better is no indication that this is a treatment for COVID or that it's a prevention. That's why people do clinical trials that are double blind, right? So it's irresponsible to go around and post videos about treatments that have not been tested, because they give people false sense of security. And moreover, you could have conditions and be taking medications where eucalyptus oil and some of these other things are not indicated. And of course, you're going to take it without consultation with your doctor. So one has to be very careful. I mean, think about something as simple as grapefruit juice. There are lots of medications that if you're taking them, they tell you do not drink grapefruit juice. As simple as that. And so you have to be very thoughtful about what you take. Again, I'm saying anything that you can eat or drink or do that will boost your immune system will absolutely help you with any disease. Not just COVID, but the flu and other things that you could pick up. It will definitely help. But none of these things are a preventative mechanism, or will they cure you of COVID, because we just don't have that at this point.
We have two more questions before I hand over to Pastor so that he can close up. One says, One says, if infected, would the vaccine be a method for curing or just prevention?
That's something that I'm sure that they're going to figure out as the post EUA post authorization and more people start getting vaccinated. When you have tens of thousands to millions of people getting vaccinated, you learn more about the vaccine than you did in the clinical trials. And it's one of the things that I'm sure that they will be looking at, that, you know, if you actually have COVID and you got the vaccine within a certain period of time, does it reduce your symptoms? Does it keep you out of the hospital? I'm sure that they'll be looking at that. But that would be good, wouldn't it?
Last but not least, our last question says, do we need to be concerned about false negatives or false positives? And do we all need to be tested?
Yeah, this is one of the great failings of this pandemic. Testing should have been put in place that any and everybody who needs a test got a test. Let me give you an example. I taught a course, a lab course, face-to-face lab course over the fall semester at the University of Massachusetts. And how were we able to pull this off? I had 30 students in my class. We met 3 hours at a time, twice a week. But my students got tested twice a week, every 2 days. They got tested for COVID. I got tested once a week. I still get tested once a week, right? With that, I had a single student in my class who became COVID positive. They caught it before she was able to transmit to me, my TAs, or anybody else, even her lab partner who worked closely with her. Why? Because the testing was in place. If testing is in place, we can have a handle on a pandemic like this. Without testing, you don't know who is carrying the virus. My student never showed a single symptom, not even a little sniffle, but yet she was positive, right? So can there be false negatives? I'm sure that, or positives, I'm sure they can be. But you have to remember what type of test this is. The testing that is being used for this virus is a PCR test, right? It's actually a real-time PCR. In other words, what they're doing is that they're getting that RNA that you find in the virus, and they're specifically amplifying it, and then detecting this amplified RNA in a machine system. This is the most accurate test and the most sensitive test you're probably ever going to get for anything, right? And that's why it's pricey to set up these testing. You know, this is an amplification. In other words, if you, let's say they were just 2 viral particles in the sample that was taken from your nostrils. This machine, in less than 20 cycles, would be able to amplify that to millions of copies that can readily be detected, right? And that's the strength of it. That's why those quick tests, like the ones that they had at the White House, could easily miss people because they weren't designed to be used as diagnostics like this. It's way too quick to go through this amplification. So there are tests out there that are not as sensitive as these qPCR tests, quantitative PCR, real-time PCR tests, that are the norm. This is the test that I get every week. It's the test that my wife gets every week. That it's very sensitive, very accurate. And those are the testings that you want to get.
So that was our last question. So before I hand off to Pastor, I just want to say thank you so much, Dr. Webley, for coming here and speaking to us. Thank you for taking our questions. I think I speak for everyone when I say that your students are really lucky because you're a wonderful teacher. And so I'm going to go ahead and hand over to Pastor so that he can close up for us.
Pastor, there's a member who's on an iPhone with their hands up. I'm not sure if they had a question. I said want to leave. This person had a hand up. I'm not sure if it was by error, if they got their question answered. An iPhone. Okay. All right, thank you. Go ahead.
Okay, I'm not seeing the person. Ashley, did you see that person?
Yeah, it's on my screen. It just says iPhone. That is up in it's the first one on my screen, but of course it's randomized on everybody's screen.
Okay, well, I think we're going to bring this to a close anyway, Doc. I want to join with Ashley in thanking you for an excellent presentation to us this evening here at the Lehigh SDA Church and our friends who join us from around the country. I want to thank Elder Paul Mears for his recommendation to bring you in. And certainly we are justly proud of you and for your presentation. I want to thank our audience for paying attention to this forum and therefore showing up with such wonderful presence. This is not a recommendation to take or not to take a vaccine. These seminars that I will have, and there will be 2 more, is designed for our education and for us to make what I refer to as an informed decision on an individual and personal basis. I believe a church body such as we are need to present what we determine to be credible information and scientific information. We need to make that available to each and every one in our church community. And so that's what we're doing. I will bring in a few weeks a medical doctor in to get a perspective from, and also a researcher. And I'm also going to bring in a patient who was a near-death patient that recovered, that you all know, in the person of Dr. Alanza Smith, to give his experience as real-life, real-time experience, and to emphasize the reality of this illness that has plagued us. And so that will happen on January the 9th. It probably will not be a forum. It will be a divine service and through the day activity, because that is our prayer and fasting day. So we're going to contour that to take part in the early section of the day and give much time to that for January the 9th. And then after that, we'll bring a physician in. I think by the time we get around to April or thereabout, most of us or many of us might be able to get the vaccine. When I say that, I mean the general population after the first responders have been vaccinated. You've given us excellent scientific clarity this evening. We will have to go back over it and look at it. I imagine that we will have some of your presentation available to us. If so, you could just send that to Ashley or to myself, and we can have another view of your slides to get some refreshing as to some of the items that you did address that we have not yet fully assimilated. So thank you so much, Dr. Webley, a graduate of Northern Caribbean University, I must remind you. And these presenters from our October extravaganza that we had in the forum have all been people that are people like us, that look like us. I noted you mentioned that Black men are suffering mostly from this disease in terms of death. And that to me is something of note for us who are from the Black community. I do also know that as of today, we have probably 1 in 1,000 people dying from this disease. That is a grim number for us to face. We have about 330.75 million people in the United States of America, and to have 1 in 1,000 dying, that's not something to sneeze at. We are at a number now of 331,000 people dead. And with what is going on with respect to travel, we will be at 400,000 by the 3rd week in January. This pandemic is not going away right now. And so we have to be careful with our use of the masks and the water on the hands, washing and just taking care of ourselves. COVID is circulating around us in different ways. We are not definitely immune from it. And we have churches who have losses. There's one church I know of in another state that had 17 people sick from COVID, and one has already died and another near to death. And just because they have been careless, very careless, they have not presented it to their congregation as something to be careful about. And so they have opened and they have been running church and then allowing everybody to fall sick. And it's a small church at that. So that's very irresponsible. Lehigh is continuing to try to be responsible, and we're giving us the information for us to take very good, careful look at. As far as the myths are concerned, I'm thankful that you began at the top to give us some idea of how we can debunk those myths. And I guess that's one of the things I'd like to go over and look at. And also the care that we need to take in looking at WhatsApp as our news disseminator, as what we would think is authentic news. And I think by now, most of us who are critical can discern that not everything on that platform is information that we ought to be hurriedly embracing. So thank you, Dr. Webley. Appreciate it once again. Thanks for coming. For all our visitors and guests, I'm going to take a quick run through. I see Dr. Rutherford. Again, thank you for being here. I know my brother and his wife were on. It's a lot of us on this evening, so I'm going to miss a lot of you. Some of our Sabbath school people that are on every morning with us in our class, Stella Troy is on. I see different names that I do not recognize. We just want to say a big, hearty welcome to each and every one of you. And we invite you back when we have another forum coming up shortly, and indeed to the service on January 9th. Some of you may have heard Dr. Lanza's experience already. I want to bring it full face focus to our church in the context of this presentation that we got this evening, to look at a recovery situation, but what he had to go through in order to recover. And I thank Veronica and Tanya from up the south and for being here as well, and all those who I haven't pulled out here, the Burks, I guess I did say before, Sister Evangeline and Dr. Tiff and so on from around our Sabbath school class era. God bless you. Have a great evening. And I'm going to bow in prayer right now. And I think maybe Genevieve might come back in with prizes. Let's pray. Heavenly Father, I thank you. Thank you so much for the intellect and for the exposure of knowledge that you have blessed our people with. And I thank you in particular for having blessed Dr. Webley with his expertise that he could share with us this evening, specifically noting the coronavirus pandemic and its nuances, its efficacy in terms of the treatment that is being provided by Pfizer and Moderna and others that will come. Thank you for giving us our mind, the ability now to explore in our thinking what our personal decisions would be and could be. And I pray that your Holy Spirit might direct us and bless us and lead us in the right path, so that we can also be cognizant of the fact that you are the God, you're the great physician, you're a creator, you're a redeemer, and we depend solely on you for our soul salvation. We thank you for that assurance, and we receive it, we embrace it in the name of the Father, and of the Son, and of the Holy Spirit. Amen. Thank you.